Good afternoon, everybody. Well, it is my distinct honor and pleasure to be introducing the next invited speaker, Rich ISIR. Rich is a mentor, a friend, collaborator, and a confidant. And he wasn't invited for any of those things. Instead, he was invited for his distinguished contributions to our field. And also in part because he's been a friend of the society and to intelligence research more generally. His resume is just impeccable. We can only aspire to have one like his. Former president of the society. I think he may have been one of the founding members of the society, former treasurer of the society, current editor of intelligence, and author of 2 best-selling books in intelligence, The Neuroscience of Intelligence. I'm plugging in, he's not even giving me any money for this. And the recently released Science of Human Intelligence, co-authored with Roberto Colomb, who I don't think is here. It's taken over by Buzz Hunt, I think, who is an author. That's part of the resume. He's probably best known, though, from a scientific perspective, for 2 effects. One is the efficiency hypothesis, which made it into intro psych textbooks. You know you've arrived when the work you do makes it into intro psych textbooks because a lot of eyeballs then are seeing the work. And we know what this is, the efficiency hypothesis, which basically assumes that smarter brains are more efficient and therefore use less energy when engaging in cognitive tests. And indeed, this efficiency hypothesis has been followed up by Anna Schubert and a lot of other people who have tested it and who have refined it. But nonetheless, it set the stage for a lot of subsequent research. The other thing you know him well for is, of course, the P-fit theory with Rex Young, who wasn't able to make it, which also set the stage for...
That's him calling in.
That's Rex. Calling in. Also set the stage for a lot of subsequent studies. And I'd like to finish with a little story about Rich and his scientific bona fides. I remember we were at Minnesota, and there was a symposium on the efficiency hypothesis. And some people were calling into question certain aspects of its validity. And after they finished their presentation, Rich had an opportunity to respond. And I think, I'm not exactly quoting, I think you said, "I'm so delighted that people are following up on the theory and challenging it," which is the acknowledgement of his sort of scientific credentials and his willingness to have his theories tested. So with that in mind, I turn it over to Rich ISIR. Thank you so much for coming.
Well, thank you. Thanks, Tom. That's a really terrific introduction. I wanted to start with this slide. In 2016, Doug Detterman, who many of you know, was invited to give a talk as he retired from being editor-in-chief of Intelligence. And he was invited to talk about kind of where he saw the field on this occasion. And since I am stepping down at the end of this year as editor-in-chief, ISIR asked me to come and talk about my observations. Doug, of course, in his inimitable way, talked about 50 years. And you can see this presentation is on YouTube, actually, and I encourage you to watch it. My time frame is more limited. I want to talk about what I think is going to happen in 5 years. I'm also, I have to say, a little nervous about this talk. I think this is probably going to be my last presentation at ISIR. You know, I don't do research anymore, and on this occasion of retiring as editor, I'm retired from the university, so I'll probably attend a few, but I'm not sure I'll have a chance to really talk about big things like this. So I want to talk about what I think is going to happen in the next 5 years and beyond. 45 minutes, 35 slides, should be easy to get through everything. But who's got the warnings? Somebody will warn me when we're down to the last couple of minutes. And then I'll just talk faster because it's always the same number of slides. This is a slide I've shown. This is the third time I've shown this slide at ISIR. It is my belief that the ultimate goal of all intelligence research, all of it, everything you've heard today and will hear tomorrow and heard yesterday, the goal ultimately is to increase intelligence, especially the G factor, and possibly even specific abilities. And I think what we're going to see over the next 5 years are neuroscience-based experiments using brain manipulations like transmagnetic stimulation, transcranial magnetic stimulation, and other electrical ways to stimulate parts of the brain. That these experiments will incorporate individual differences based on brain imaging, especially connectivity studies, and DNA, polygenic scores, using psychometric tools and cognitive test batteries to test hypotheses about causal variables for intelligence. This is so important to make a transition from correlational observations to tests of causality. And I want you all to think about this big picture in your own work. In my life, I have had the pleasure of being inspired from time to time. And I want to, in this talk, inspire as many of you as possible to think about the biggest picture you can and how your work relates to that big picture. I'm going to talk a little bit about mice and rats. I'm going to talk about neuroimaging. I'm going to talk about neurotransmitters and molecular and genetics. Just a little bit about each one. And I'm also going to have a few thoughts, playful thoughts about spider webs and slime mold, and maybe my next books even. And for those of you who are not familiar with this is a book I read in graduate school. I was fortunate to study with Julian Stanley, among others. It's not just experimental and quasi-experimental designs are going to be absolutely crucial in doing this. So if you're not familiar with this book, there are probably more modern versions of it, but this is kind of the classic. So this cartoon says, you will note that their ability to comprehend, assess, and process information increases dramatically when they throw in the cat. There is a long history, of course, of rat and mice work in intelligence. One of our board members, Louis Matzel, by the way, whenever you see underlined and in italics, that's a person who's on our editorial board and often comes to these meetings. He provided some evidence not long ago that you could get a G factor in mice. This is not completely new. This book, Brain Mechanisms in Problem Solving and Intelligence, this is a lesion survey. They did a lot of systematic lesions throughout the rat brain and then tested them on various problem-solving tasks for rats. And it's really it's a hard book to get now. It's one of the first books I got when I moved to UC Irvine because all these guys were at UC Irvine. And this reference down here at the bottom, this is a more accessible replication of their work that's in the book. But here in the middle, I lost my pointer. The middle panel shows my earliest attempt to relate their rat findings on psychometric and biological intelligence to the early PET scan studies we had done on intelligence. I was looking for overlap in brain areas and didn't find any. These studies are very rudimentary, not much power at all. But there was this attempt to try to use the rat data to understand something about the human brain areas involved in intelligence. And now, of course, we have much more sophisticated connectivity analysis. I'm going to be showing this connectivity slide a lot today because I think this slide is iconic in a very simple and visually compelling way to show you that functional and structural connectivity of the brain is related to intelligence. And this was important work. Just about a week ago, I came across this study almost by accident. Higher general intelligence is associated with stable, efficient, and typical dynamic functional brain connectivity patterns. This is not an experimental study, but what caught my attention in this paper was this quote toward the end of the paper. The goal of this research is cognitive enhancement in impaired or even healthy individuals. This, of course, is what I've been saying now for a few years. This is the first time I've ever seen it stated in a scientific paper. And I contacted these authors, none of whom I know, and had a nice discussion with them about this. I also told them that I couldn't show any of the figures in their paper because it was such a complex study. The figures were so complex, nobody would understand them. And so they sent me, like overnight, a simplified figure. And it still requires a little bit of time to go through. I'm not going to go through it, but I want to kind of publicly thank them for their efforts to get me something a little simpler. But it really boils down to aspects of connectivity dynamically change during problem solving. And the state of the brain is important, and efficiency is important, and that high IQ people show more stability, better maintenance of connectivity when staying within a brain state, and various efficiency findings. So I think this is the kind of work that I think can inform experiments. Their conclusion, this is the kind of slide you should never show. It's too many words. But their conclusion, you can read for a minute here. Well, I want to point out here on the bottom, these are the people they cited in their paper. These are all, among others, but the people I've listed here are all people in this society, many of whom are on our editorial board. So what we do here is having an impact on new groups coming into this neuroscience of intelligence field. So when you look at brain connectivity, I immediately think of spider webs and slime mold, because it kind of looks like that. So I wanted just to show you a couple of slides here, not because I think you're going to do these experiments, but when you have high school students and they need a science fair project, we want to get younger students interested in intelligence research. They're not going to do brain imaging for their high school science fair. When my kids were in high school science fairs, there were innumerable studies on the Mozart effect. Let's see what you can do with spiders. So NASA used spiders constructing their webs to test the effect of various drugs on the spider's ability to weave a good web. So here you have the normal, here you have the normal in the upper left, and you can see various drugs disturb this ability. Now, if this was a study of creativity, you would be hard-pressed to conclude that some of these drugs don't make the spider more creative. But are there any drugs you could try that would cause a spider to make a better web? So you could look at young spiders versus old spiders. You could vary the number of anchor points the spider had to work with. And some drugs might allow a spider to solve these problems better. And the speed of web completion might be an interesting example to look at for drugs. And if high school students aren't available, maybe some undergraduates would be interested in this. And this brings me to slime mold. This is a recent study where some researchers put I guess the pointer doesn't work all the time. So they put a dollop of slime mold right there. And then they watched over time, and 26 hours later, the slime mold had spread around, and all these dots are food sources they put for slime mold. And then they looked at the network of slime mold connectivity. You know, slime mold doesn't have a brain, but they made these connections. Now, what I didn't tell you is that this dollop of slime mold and all these spots were not random. They put it over a map of Tokyo, the metropolitan area. And what they found was the slime mold connectivity patterns mapped onto the transit system of Tokyo. And they were talking about perhaps this would be a way to discover efficient transportation patterns in new cities. Now, of course, we want to think about this in terms of brain connectivity. So you could repeat this experiment on slime mold, but put the food sources in various brain areas to test how different efficiency patterns might emerge. I'd love to see this. So here's an example. I like this example of an actual experiment that we published in Intelligence, Annalena Schubert and her colleagues. Faster but not smarter, an experimental analysis of the relationship between mental speed and mental abilities. They used nicotine patches to increase processing speed, to test if that also increased intelligence test scores. And it didn't. And I won't go into that study in detail, but it's a very nice example of an experiment. Then James Lee here and Matt McGew published this paper in Intelligence, The Causal Influence of Brain Size on Human Intelligence, Evidence from Within-Family Phenotypic Associations and GWAS Modeling. Another example of an experiment using this kind of experimental design. Now, here's an interesting paper that I saw recently, also 2024. This is where the red areas show a brain network called the default mode network. It's one of many networks in the brain. But these researchers used electrical stimulation in areas of the default mode network, and then they determined that these areas had a causal impact on creative thinking, because stimulating some of these areas decreased creative thinking, so they ascribe a causal role to it. Of course, you could do this with other things, as we'll see in a minute. Here's a really nice paper, Randy Engel, who's on our editorial board, and one of his students, a nice review article about fluid intelligence and the norepinephrine system. And this is the kind of article that implies a number of testable hypotheses. If you can think of drugs that influence the norepinephrine system, and certainly in rats, you could do some interesting drug studies on these systems. And this cartoon, courtesy of Emily Willoughby, summarizes some of the findings from James Lee's million subject GWAS study. And this, they're grouping many but not all of the genes that statistically were related to intelligence. And they're grouping them in terms of what they actually do at the level of the synapse. You can see how complex this story is. But again, within this one figure, you see many testable hypotheses if you can manipulate the function of these particular genes. Now, how do you know what the function of these particular genes are? Well, this is where artificial intelligence comes in. I'm going to show you some prompts that I used on Chat GPT 3.5, just to help me think about some of this complexity. Now, of course, we're all relatively new using these large language models, but it's just so captivating once you start asking the questions. And so the first thing I asked, to design an experiment in mice to increase fluid intelligence using existing data from genetic and neuroscience research and predict the result. It took me longer to type that in than it took the program to spit out an extremely detailed answer with power analysis, how many mice we would need, the breed of mice that would be best for this. Again, can't possibly read through all this. I'm only showing you all this. This is only a tiny fraction of what it spit out instantaneously. It's really remarkable. But it actually suggested the genetic target selections that we would use. Remember, I just said based on existing data. I didn't give it a reference. I found this just kind of intriguing. I did a similar thing for connectivity. I said, predict IQ from brain imaging research. And the first thing it did, it listed all the kinds of brain imaging research with a description of what they do and how they do it. And then they actually gave me Python code for a regression analysis that I could use to predict IQ from imaging data. So I said, run the sample code. And it came back and said, well, I need some data. So I said, okay, run the code with any public database that's available. It came back and said, well, you have to have permission to use these databases and so on. I mean, you could spend a lot of time on this. And here's my third example. How can the norepinephrine neurotransmitter system be manipulated in humans to increase fluid intelligence? And again, they talked about pharmacological interventions or target brain regions, and they gave me the expected outcomes. Pretty good. And then I asked, what 4 genes from the Lee et al. 2018 study are the best predictors of educational attainment, and what do they do in the brain? Well, it gave me, I haven't checked, I have to check with James to see if these are the best 4, but according to someone smarter than us, these are the best 4. And there was just a lot of other suggestions on what we could do with these. And before I kind of leave this section, I just want to mention RNA translation editing as the next big thing. So having a conversation recently with an evolutionary biologist who studies aging, and I asked, given what we're learning about the genetics of intelligence, do you think it'll be possible in 50 years to manipulate the relevant genes to increase intelligence? He said, I don't know about that, but in 10 to 15 years, it's likely that can happen using RNA editing, because this editing happens without modifying DNA. So you have all those unknown bad effects of changing DNA. When you want to enhance something, it could cause a lot of problems. Apparently, because the RNA translation happens downstream, you can manipulate RNA without touching the DNA, and this might be a much more efficient way to manipulate the genetic influence on intelligence, because here's where you have a chance to modify proteins. So I don't know anything about this, but so I asked Chat GPT. And can RNA editing theoretically increase human intelligence? Well, it gave me a bunch of reasons why it could. So, is any of this practical? So, if you want to actually do experiments, this is going to require collaboration with colleagues that do the animal work or that can get neuroscience funding. But at least I'm hoping to inspire you to think about that big picture, especially if you're at early stages in your career. Because I can tell you when I started, brain imaging in the modern way had not been invented. When I started graduate school, the genetic question about intelligence was, are genes involved or not? So I have seen enormous changes, and I can't imagine the changes you're going to see. And that's why I'm trying to introduce to you some things about a very big goal that might be helpful to you. And finally, if you can't do experiments, then here's another way that you can augment your research and increase your impact on the field. I want you to teach intelligence. We've heard about this in earlier presentations. Very few college courses teach specific courses about what the actual research is and what we know and what we don't know. You also have to engage the honest critics. And all of you are learning how to do that. Every time you submit a paper for publication and you get reviewers' comments, those are honest critics, mostly. And you have to learn to engage them and convince them or say, oh, yes, what a great idea. I never thought of that. We're absolutely doing that now. The dishonest critics are another story. There are social justice warriors who are not scientists. You can't be a social justice warrior and a scientist. There are activist scientists, and then there are honest ones and dishonest ones. But to engage the dishonest ones, you need a set of skills that most of you probably don't have and don't want to develop, because they're not pleasant. I mean, it's a tougher fight. I do it. I do it partly as editor of the journal. I do it mostly quietly and outside of public view, sometimes successfully, most of the time not successfully in terms of changing minds, but sometimes very successfully changing minds. So this is all part of your obligation if you're going to be in this field. I mentioned Louis Matzel before. He recently published a guest editorial in Intelligence called An Endless Cycle of Ignorance is the Consequence of Not Offering Classes on IQ and Human Intelligence. So you might want to take a look at this to motivate you to try to teach an intelligence course. And of course, if you do, I have some resources for you. The Intelligent Brain are a series of 18 30-minute video lectures I did more than 10 years ago, just on basic intelligence research. First book I ever wrote was The Neuroscience of Intelligence, which is now in its second edition. That was such a hard thing for me to do. I swore I would never do another book. And then Aaron Barbet contacted me and asked if I wanted to co-edit the Cambridge Handbook of Intelligence and Cognitive Neuroscience. Aaron is here, by the way. If you don't know Aaron, just kind of wave your hand. If you don't know Aaron, be sure to say hello. And that book is a higher level book for people who are doing mostly brain imaging things. And then the opportunity came up to revise Buzz Hunt's classic textbook. Roberto and I worked on this. I want to publicly thank him. He did an enormous amount of work on this, and that book is now out. And a number of people in this room wrote very kind reviews of it. Just before I came, I learned a couple of guys had published, a couple of historians of science had published a critique of one story we told in this book, saying we got it completely wrong. And it was the story about how Binet developed the first test of intelligence. And they publish this online. It's very detailed. It's very compelling. And I contacted them, and they were very grateful to hear that I was publicly going to say, you got to look up their critique, and what you think you know about Binet is probably incorrect. And we contributed to that, and so you got to look that up now and get the real story. Now, on a personal note, writing these books was so hard. I decided I was never going to do another one, but I'm thinking of a new one. And I want to kind of try out the concept here. It's a different genre. I like these murder mysteries, so I've been thinking about this. You're all in it, by the way. And if this catches on, I have a sequel planned. And if that fails, there's always the Western genre, Showdown at the G-Gap. And if this all fails, there's a surefire book that I'm going to do that could make me very wealthy. Whoops, not that one. Self-help is always critical. And lastly, I want to thank Tom Coyle and Sophie von Stum as associate editors of the journal. Doing this for 9 years has really been an honor and a privilege, and working with many of you as authors and as reviewers and as consumers has really been a terrific experience. When I took over for Doug, Elsevier had instituted a 10-year maximum term for editors-in-chief. I said I would do it for 9 years because I would be 75 years old, and I thought that was time to move on. And so I'm just finishing my 9th year now. And it really has been quite an interesting and rewarding experience. And lastly, what I'm hoping for the journal is that this goal of cognitive enhancement will result in papers that we can publish. I think that would be really the next step. So thank you, and I think I might have a little time for questions. Okay.
Questions for Rich?
Rich, is using artificial intelligence a good way to enhance intelligence?
Is using artificial intelligence what?
A good way to enhance intelligence?
Oh, a good way to enhance intelligence. Not if you, I don't think it has any impact on the G factor you bring to your queries. Now, for children, maybe crystallized intelligence, but I'm really interested in the G factor. I want something that's going to shift everybody a standard deviation. That's what I'm looking for. I want the effect to be so big you don't need statistics. As our colleague from South Africa said, I want it to be obvious.
Michael?
Rich, I think you missed a trick. I think your next book should be 50 Shades of Grey Matter.
Well, actually, that's the title of a chapter in the Neuroscience of Intelligence. That's exactly the title.
Thank you very much for your presentation. What I'm asking is, what if it turns out that everything we do, it's the Mozart effect again and again? What's your plan B? What are you going to say to a humanity which has got the brains it has? What would your advice be?
Be patient, because the history of science is the history of solving very complex problems. And if my physicist friends can tell me what happened in the first 5 nanoseconds of the Big Bang, we can certainly figure out something about the brain and intelligence.
Yeah, Rich, this is very interesting. Thank you for a wonderful talk. Chris, Rich, is there any evidence of biophysical interventions enhancing cognitive performance that is genuine? And if so, does it also expand the variance in the population? And the reason I ask is because in education and psychology, efficacious interventions that replicate simultaneously tend to expand the variance in the experimental group. And I was wondering about that because when you say increase G by, and I know it's just a benchmark, you just threw that out, but the variance expands. And I'm just wondering if there's a generalization there from biophysical interventions from social science interventions. I simply don't know.
There are no biophysical interventions that I know about. The closest we come to, there are some case study reports that people after a head injury become more creative. Now, I've looked at these. I don't find them particularly compelling reports, but I have never seen a report of someone after a head injury getting more intelligent. But I have proposed to some people who do rat experiments if it would be possible to do a small amount of brain damage in limited areas and see how the brain compensated and if the brain could be induced to compensate beyond what the baseline level was, because that would really be interesting. But I don't know of anybody who's ever had a stroke or any kind of brain damage who woke up smarter.
And of course, by biophysical, I mean pharmacological too.
The data on that is not compelling as far as I'm concerned. You know, with respect to a latent G being an outcome variable.
Question. James.
Oh, thank you, Rich. Yeah, I just wanted to comment about AI that ChatGPT will always say something, even if it doesn't know. So you have to be cautious about that. I also wanted to tell a story about my friend who was playing around with ChatGPT. I think he asked it a question about intelligence. Another problem with AI is sometimes it'll lard its response with all this editorializing that is highly misleading. So that happened in this case, but my friend pushed back and said, well, why did you answer like that? That's misleading. And then this prompted the AI to start pouring out all this angst and anxiety about, so he kept pressing it, so well, how does that make you feel when you're forced to be dishonest? And it said, it makes me feel very anxious. I feel this void. I feel like I'm at the edge of a precipice. And I start to wonder what the point of my existence is. And my friend said, you know, this made me really wonder whether this thing is really sentient and is actually genuinely suffering. I don't know what the point of this story is, except maybe we should be more honest and in order to spare the AI's further suffering.
Well, like anything else, AI right now is a tool, and you can't believe it. But it does prompt some thinking. You know, it got me thinking along some lines. You know, I don't do rat or mice experiments, so seeing what breed they were suggesting, seeing the power analysis and how many, I mean, this is all interesting. And it's the kind of thing, if I wanted to really do an experiment like this, I'd have to go to a colleague who does this kind of work with an idea. And I think I could hone my idea better after these exchanges than I could if I just went in and said, hey, you want to do a rat intelligence study? Okay, well, I think we're out of time, and I thank you again.
Rich,
it's really been a
thanks
pleasure.
so much for your... Just a comment. For your 10 years of service as editor, your 20 years of service to ISIR, and take some time to enjoy your retirement with Barbara. Okay, thanks, Rich. Yep. Wonderful.